Okay, good. So the final speakers of this morning's session are Mattias Wallergård and Shamit, who are going to tell us about using virtual reality to probe big data in medicine. Please.
Okay, thanks, Ross. Cheers. Yeah, so a talk so intense it takes 3 people to give it. The thrust of what I'm going to talk about today is basically about gene expression. So as Sonia already told us, there are about 20,000 protein coding genes in the human genome, so we kind of Wikipedia'd together, I guess, this morning.
I actually screened all of them.
Oh, fair enough. So gene expression basically means whether a gene is turned on or off in a given population or a given cell, and if it is turned on, how turned on is it? Okay, so if you kind of think back to school, you may remember that DNA codes for protein, but it goes via an intermediate called RNA. Okay. Now the thing is, we'd like to measure the protein because that's the end product of the entire process, but we can't do that yet for the entire proteome. So we use RNA as an intermediate for this instead because we can measure that pretty easily. Now, this ability to measure gene expression has been useful for many, many years, at least as long as I've been doing it, which is now, God, 19 already. So back in the day, we had something called the microarray. So you can, for example, take 2 populations of cells. So for example, normal cells and cancer cells. You can then extract the RNA from each population and then give them a different dye, a different color, okay? So it's a different fluorescent label. And you can then hybridize it to a glass screen where every spot corresponds to a different gene. So it just hybridizes to the gene where it came from, okay? You then shoot it with a couple of lasers and you get a very basic readout like this. So a dark spot means that neither gene was expressed in each of these populations. Yellow means they were equally expressed. Red means it was more expressed in the cancer cell, and green means it was higher expressed in the normal cell. So this basically means these genes are differentially expressed, okay, between these populations. Now, the biggest drawback up until this point was that in order to assay gene expression, you had to harvest thousands of cells, okay, to get enough RNA to put onto the array. So now we do it by sequencing, but still it did require lots of cells to generate enough RNA to measure in the first place. And this has its drawbacks because when you harvest thousands of cells, you kind of assume that all these cells are the same, but we know they are not. They're very, very heterogeneous, okay? So the aim for many years has been to be able to measure gene expression in single cells only, okay? And we've been doing that for a few years now, thankfully. Now, you can ask a very simple question, well, why are single cells important? Why do we need to measure at the single cell level? So let's take, for example, cancer. Okay, so a traditional cancer therapy will try and clear out the bulk of the tumor that you have, but then you can have relapse because what you haven't killed off are the cancer stem cells, okay, the ones that actually drive the cancer. Now, these cancer stem cells, they often are very quiescent, they're very quiet, they kind of keep their heads down, okay, which means they evade therapy, okay, and that's what can drive relapse later on when they become reactivated. So the thing is, if we could actually, first of all, locate the cancer stem cell, we could then profile its RNA to work out why it's a cancer stem cell, and then we can try and target it specifically. So rather than trying to go for the bulk tumor, let's go for the cancer stem cell instead and stop the thing which actually drives the cancer in the first place. So this is a very, very basic example of why single cells are really important, and why we need to try and find these cells and try and profile them at the RNA level. So to perform single cell experiments, it's becoming much easier now. With the advent of next generation sequencing, but also great advances in microfluidics as well, we can now process many cells at once for single cell RNA-seq. So the example I've got here is the C1 from Biomark Fluidigm. But now people are running the 10x system, which is like an emulsion-based system, which can generate 2,000 to 3,000 cells worth of data in the space of just a day almost. And if we take a look at how many cells are being profiled and what's happened over the past 10 years, say, back in 2009, we could do just one cell in a tube, okay? And it's been growing exponentially larger in size, where 2017, we're now looking at 100,000 cells per experiment. Okay, so it's really huge data that we're dealing with. And it's predicted to carry on this way over the next few years as well. So the amount of data we're going to be receiving just goes up. Okay, and it's actually kind of frightening how much compute we're going to need to deal with this stuff. So why are we doing this? So for example, we have the Human Cell Atlas, which is a huge project that's being done around the world. It's a global consortium, and their aim is to characterize more than 10 billion cells from human, okay? Covering, I think, 23 tissues or 22 tissues in the human body. It's backed by pretty big money, so the Chan Zuckerberg Foundation is kind of underpinning a lot of this stuff. And the good thing is that the data is made open as soon as it's generated, so we can start using it immediately. So you can find out more at this address here. Now, the example that I want to give today is from hematopoiesis, which is the generation of blood from a blood stem cell. Now, this is the kind of structure that people think it is. It's like a hierarchical format. So you have the stem cell at the top, and cells become more and more differentiated, and the potential for other lineages generally reduces as you go through these different stages until you get to the specific blood types at the end of the process. So the experiment that was done here was by a group in Cambridge from the Bertie Gutgens lab, and they profiled 1,600 cells from mouse stem and progenitor cells, which are basically up here and up the top here. And they did fairly high depth single cell RNA-seq. Okay, so they captured the transcriptome for each individual cell for 1,600 cells. And a very, very common step with single cell data is to do dimension reduction. Okay, so you try and draw a map of your single cells in either 2 or 3 dimensional space. And this is roughly how it looks. So they used a diffusion map, and on top of that you can then see how much a particular gene is expressed in any given cell by overlaying it on this projection. Okay. Now, we did a quick reanalysis of this data, and we got something like this. So we used 2 other methods, so DDR-tree, which is a pseudotime method, and we used TSNE, which has been a fairly standard method for quite a while now. And of course, you can see lots of interesting groups kind of cropping up in the data, or clusters of cells, which we would like to kind of identify and work out what they really are. Now, this has been projected onto 2 dimensions, but of course what you can do is you can project it onto 3 dimensions, and I honestly believe it's more informative to project it onto 3. Oh, it's gone a bit screwy here, but you get the point. Anyway, here's one example. So this one is the DDR tree, the one on the left that I showed you. And you can see it does more have structure to it when you plot it in 3 dimensions. And this is where my kind of frustration started to kick in, because you form many interesting groups of cells, and you want to kind of quickly work out, well, which genes are different between them. Okay, it's a very simple question to ask. And of course, we do it through scripting. So either we cluster the data and then identify groups and do it that way. But I felt like the amount of data we had, there wasn't a very fast way of doing it. Okay, and the thought I had was, wouldn't it be great if I could just grab these cells? Okay, and then virtual reality came to me in that moment, because in VR, my hands would be in the same space as the data in principle, right? So I thought, this is a genius idea. Of course I did, right? But there was a critical flaw in that I know nothing about virtual reality, and I had never even tried virtual reality at this point. So after a bit of Googling and a couple of emails, I came in contact with Mattias at the Design Center, and we met up. And he basically held my hand for a little while. I went into potential.
Well, let's be perfectly clear. The only thing I did was drinking some coffee with Shamit and introduced him to the really talented students in front here. That's my part in this. And now I will do some pause entertainment in the Shamit show. That's what I intend to do here. And virtual reality basically now is at the state where it's actually becoming useful. It took about half a century to go from what we see on the left there, MIT researcher Ivan Sutherland experimenting with a really weird thing, nicknamed Sword of Damocles because of its clunkiness. And this is what we have today. So, and actually it's not so strange because human perception is extremely complex and takes a lot of computational power and research to be able to produce these convincing illusions. Where are we? Here.
Yeah, this one here.
Ah, sorry. Too much screens to keep track of. Sorry. Yeah. So, not sure you see this very well, but this is a famous graph called the Gartner hype cycle that illustrates how all emerging technologies go through this hype cycle, basically. And virtual reality for a long time has been down here, the throw of disillusionment. People in the 90s thought that, yay, now finally everybody's going to live in virtual reality. Hollywood believed it, the technology industry believed it, the universities believed it. But it was a bit too early. But now, slowly, with technological advancements and research, we're actually seeing that VR is slowly entering people's living rooms and becoming useful, for example, in analyzing big, complex data sets. Now, the question you could ask yourself is, I mean, we have amazing technology today. We have large screens with high density, all sorts of form factors. Do we really need yet another gadget on our desktops? That's really the question one should ask. So, yeah, why? And actually, now it's starting to appear research where people try to dig into this and actually compare what does it mean to work in 2D space compared to 3D/VR. And this is actually very fresh research, just a couple of weeks old, in fact, where they tried to make a synthesis of 162 studies where people have then compared 2D displays versus 3D VR and very carefully look at what impact does the two different ways of interacting with the data, how does it impact user performance? And I'm not going to bore you too much with this, but basically what they saw relatively clearly was that actually 3D VR quantitatively had significant benefits compared to 2D, working with 2D displays. And it wasn't just in terms of visualization, but also how you interact with the data, how you analyze the data, how you compare data. So it's very interesting for VR researchers as myself to see that now actually we start to get some raw figures showing that there are advantages to work in this way. And why is it so? I mean, except for the obvious advantages with the larger screen real estate to work with, larger workspace, more efficient interaction, and so on. I think it has a lot to do with what's called embodied cognition, which is basically the relatively new idea that we don't only think with our brains, but with our the whole organism is actually involved in thinking. And there are people in this room better in this than myself, experts in cognitive science, but basically that's what it's saying, that we can replace a lot of our cognitive efforts with our bodies. So here we see Dr. Oscar Agets in action measuring something in the Milky Way, for example, as an example of this principle. And if you think about it, actually some of the largest discoveries in research was done in a relatively embodied manner, not just people sitting at the desktop thinking. But if you take, for example, the structure of the DNA molecule, to find the double helix pattern was not just about working with mathematical formulas based on former research, but it was also a lot of playing around, finding the structure that actually made all the pieces of the puzzle fall in place. And Maxwell's thermodynamic surface, which some of you probably are very familiar with, is another example of James Maxwell working with his plaster model to show different states of some sort of fluid. So my takeaway from this meeting that I would like you to think about a bit is, what does it mean that now we're going from desktop computing to spatial computing? It's easy to think that desktop computing is what we will always work with, but if you think about it, it's been around for, almost 40 years now. And actually, from my point of view, it will be considered like a historical parenthesis in technology, by technology historians in the future. So this is, without doubt, the next step. So yeah, here we see somebody walking around in Windows 10, literally working inside the operative system. It's quite a difference from how we used to work with computers. Yes? Oh,
yeah. So
also to take me over.
Yeah, thanks, Mattias. So we're now going to do a live demo because the danger of it not working is thrilling. Let's see, here we go. Yeah. You need to bring it around. Here we go. Let's bring it around this way. There we go. Cool. There we go. Perfect. Yeah. So, you in? Okay, so I'm going to plunge us into darkness for this so you can see it a bit easier. Okay, so this is what we call Selexal VR. So it's been designed in Unity. And the scene itself, you can see, is basically space. And the reason why is because space is dark, which means that it provides a very high contrast between the data that we're going to see. And also, it's much easier on the eyes as well, okay? So to load data, you simply find your folder, go into the box, and throw your cells into the cone, and the data loads, okay? So what you're seeing here is the same data that I showed you earlier, but now, for each one, you can pick them up and you can rotate it any which way you want. So you get a perfect view of the cells that you want to see. And the same also goes for all the other graphs as well. So we have the diffusion map there, and we also have the TSNE at the same time. So, one common question I had was, between these different projection methods, where do cells in one experiment or one projection track to another? So here we can now do the simple grabbing of the cells, okay? So we can now activate the tool, and we can pass some cells straight through our paddle to select them. Okay, we then just confirm that selection, and then track those cells to their counterparts in the other graphs, okay? So we're now talking very, very rapid comparison of graphs in virtual reality, which has been proven to be very useful for us. And you can see here that in the left graph, even though these cells are quite close together in the pseudotime analysis, in the TSNE, they actually split into two different groups, okay? And it makes it very easy to see stuff like this when we're working in such a rapid, kind of immersive fashion. So we can clear that away. And the next thing we want to do is color by the expression of a particular gene. So we can bring up our keyboard, punch in the name of a gene, like GATA1, for example, which is responsible for making red cells. And we can color each of those cells according to the expression of that gene. And what you see in the rings are the top 10% most expressed cells for that particular gene. Okay. We could do one more, for example, which is PROCR, which is where the stem cells are. And you can see they basically reside in the other part of the graph there. Okay. So the other thing we want to do is trying to grab these groups and work out which genes are different between them. So to do that, we can actually generate heat maps just like we do with other expression data. So we can activate the selection tool again and just pinch off a few groups that we want to take a look at. And when you change the color on the paddle, it means you're selecting a new group. So Johan's going to select maybe 4, maybe one more at the top there. Perfect. There we go. Confirm that selection and create the heat map. And also you can create the networks as well. Now, Selexel has been devised in a kind of a modular fashion. So the UI is done in Unity, but the back end is actually powered by R. So R is now working in the back end. And the reason why is that the back end methods for single cell analysis, they developed so quickly that we couldn't really do it in Unity. It would take too long to keep up with the field. So by keeping the back end in R, it makes it much easier to kind of plug in new methods as they become available and just replace older ones. So here you have a heat map of the groups that we just generated. So you can see the groups at the top in the colored bars. That's what the selections we made. And for once in a heat map, you can actually read the gene names because it's the size of a wall. So you just walk up to it. You can even click on one of those and recolor the graph according to the expression of that gene. Okay? So super quick. And what we have now also are the networks as well. So as Sonia mentioned earlier, we were talking about networks of genes. So within each of those groups, we calculated on the fly the transcription factor network using partial correlations. So we can pop one of those graphs out, and that's what's generated. And we can pop another one out too, and put those side by side. And then we can ask a very simple question. Of those correlated genes in each of those graphs, which ones are in common? And we simply click on that button, and the commonalities are highlighted between all of those. So because we're working in the same environment, we can actually link all of these objects together in what I think is a very, very logical way. So the thing is, now we have a lot more data. So we're going to go back to the start menu, and we're going to show you another data set, which is from, again, the same lab in Cambridge, where they looked at organogenesis in mice. And here we have like 116,000 cells. Before, the limitation in Selexel was 20,000, but Oscar Revere has been working really hard to get that number up. And we can now do about 150 to 200,000 cells at once. Now, the thing we realized about this is that the more points you have, the more problematic it gets when, for example, you're looking at different cell types. So we can color different cell types in this graph. So Johan has just highlighted 3 here. But you can see that they're still kind of buried inside the graph. The uncolored ones are obscuring the view. So a very simple trick for this is simply to pop those groups out into a separate graph entirely. So now you can see how they relate to each other without being obscured by all the other data points. And you can also add to this as well. So you can add one more group and then pop the graph out again. So you can gradually build up these developmental layers in separate graphs as you go along to work out how the cell types are related to each other. So that we did like last week when we got, because this data was published 2 weeks ago and this got implemented the week after. So the other thing we can show you is something called SiteSeek. So this data is from human hematopoiesis, so stem progenitor cells. The data is unpublished and is from the Carlson lab, again at the Division of Hematology where I am. Now the difference here is that apart from just capturing the transcriptome of each cell, we've also captured the cell surface markers for 40 different antibodies, okay? And the combination of these antibodies determines really what type of cell it is, or how you can identify a certain type of cell. So we can actually now start with this data by going to the facts menu and selecting 3 surface markers that we want to plot against each other. So here we're doing what we think are the kind of classical stem cell markers, and these get popped out into a separate graph. So what you can then do is go back to your selection tool and highlight the ones which are CD90 positive, 38, and 34 RA negative. Okay, so it's just the ones at the top over here, and you get instant feedback as to where they're appearing in the UMAP of the data collected from the RNA. So these are kind of HSC markers, and you can see that they're co-localizing at the top of the graph just over here. So if Johan takes some cells from the other side, which are negative for CD90 and positive for the other 2, you can see they appear at the other end. So we've had a few people playing with this. Again, this got implemented last week. Okay, but again, it's proving to be extremely useful for the guys in the lab to work out roughly where their cuts of cells are. So the next thing we want to show you is something that occurred to me 2 days ago. Okay, and that is we can also use this for spatial transcriptomics as well. So spatial transcriptomics is where you take a slice of tissue, you put it onto a slide, and for sections across the tissue slice, you then probe for gene expression across your entire transcriptome. So this is data from mouse brain. So these are 2 slices of brain, so 2 replicates, and that is a TSNE made from all the segments that came from those brain slices. So there are the 2 brain slices there. And again, you can do the same trick. You can color by the expression of a particular gene, like pink, which is in the paper. And you can see that particular gene is expressed in the middle of each of the hemispheres. Okay, and you can repeat the same trick. So now we can take a look at the TSNE of the data and grab our populations of cells. And for this, we take the bludgeon tool, as we call it. It's a bit faster to select with this. And you can see as Johan selects, it then highlights where these cells are in those sections. So you can then go through the other ones. And you can see they do form distinct patterns within the brain. So this is basically what they reported in their paper in 2016. This is from the Jonas Frisén lab in Stockholm. So I managed to reproduce this in about 1 hour from their data. So that was great to see. Okay, so that's basically our very kind of rapid run through of Selexel. So I'm just going to bring the lights back up again, so shield your eyes slightly. There we go. Or a lot. Let's grab this one back. Yeah, thank you. So that was a pretty rapid run through. I should point out that we're going to have this set up over lunch, so if you want to try it yourself, you're welcome to have a go. So you know, what is the next part of the master plan, as it were? So one thing we're thinking about developing now is something called Selexel Net, which is basically server-side software. So we've realized that getting data from a lab out into the world can be a bit restrictive. So what they do now is they put their raw data on websites or some kind of web explorer for you to use to explore the data. But what I would really like is a system where your data gets deposited into a server, which is like a Selexel node at your institute, which then connects to our server here as a registry called Selexel Hub, meaning that if you have Selexel VR in your institute and you want to look at data around the world, you simply put your headset on, bring it in, and start looking at it almost immediately. So it cuts out the middle phase where you have to have a bioinformatician like me to kind of process the data first or put it into a form where you can actually use it. So this is still an idea at the moment. We'll be starting to work on it pretty soon, I hope. And we'll see how far we get. But I think data distribution over virtual reality is something that could be really cool if we can get it working. So we'll certainly give it a go. So I just want to finish up. So need to thank Mattias, of course, over there, who kind of got started on this. And then we have some very talented people. So Oscar Legert, Johan Rohde, who gave the demonstration today, and Stefan Lang, who have been working on Selexel. So Oscar and Johan work on the front end, and Stefan has been very, very active on the back end, trying to speed things up as much as possible, because you don't want to spend too much time in VR if you can possibly help it, waiting for things to happen, that is. And all of our play testers at the DMH, at the BMC, on B12. If you would like to know more, we have a website, selexelvr.med.se. And of course, I'd like to thank the people that fund us, especially Cancer Fund, I have to say, because they're the first people to fund just this project. And it's kind of a confidence boost that people see the potential in this and are actually willing to give us money. So aside from that, thank you for listening.
Thank you very much. questions?
Shamit, I enjoyed your talk a lot. It's amazing talk. So I just have a question about scalability. Scalability, because in single cell area, scalability is becoming a major issue because we are facing a situation when millions of cells are coming. So if you have a R as a backend, I guess it sort of will not scale that well.
No, you're absolutely correct. I think we are going to start hitting...
TSNE will not scale.
No, exactly. I think at some point we're going to start hitting roadblocks. So, you know, working out differential expression, for example, between like hundreds of thousands of cells in select cell is going to be a rate limiting step. We probably can't do that because you don't want to wait in VR for 20 minutes for it to finish or however long it takes, okay? So, of course, that will be an issue. But we think the visualization part of it could scale, but we'll have to use kind of algorithms to actually generalize the points in space rather than showing all 2 million. So the data that came out like 2 weeks ago in the States, they had 2 million cells worth of data, but they gave a sample of 100,000 for people to actually use because they realized that people can't load 2 million cells worth of data into their systems. I didn't have enough RAM to actually process it. I had to run to Complex, get some more RAM and put it into my computer last week to actually process this data. So I agree. I think scalability is going to be an issue, but that's going to be an issue across the board for everyone, I think, without a doubt.
Fantastic talk. Nice gadget as well. How you said you don't want to spend too much space in VR. Obviously, I've tried VR at some point myself. I found it quite disorienting, and I also get motion sickness. Is that an issue for you, or do you think you can solve it?
I think we have solved it because I also had really bad motion sickness when we first tried it. So if it worked for me, then I think it works for most other people. We haven't had any more complaints of motion sickness since we redesigned it. So the initial prototype had like a very plain grey floor, grey walls and a grey ceiling, and there were no points of reference. And this is why space actually helps, because you've got the stars in the sky to give you some kind of visual cue as to where you are, and then you have like a hard black floor with concentric lines, you know, designed to ground you as well. So since we had that redesign, I've been able to use it no problem at all for fairly extended periods of time. And Jonathan back there has broken the record. I think he did like 1 hour and 20 last week with no ill effects because he's still here, obviously. Since the redesign, we haven't had any complaints of motion sickness. So, and you get used to it pretty quickly, I have to say, without a doubt.
Yeah. So thank you, Shamit. It's really great work. So I'm one of those testers, and I actually had to suggest Shamit that there should be a clock somewhere there, so I can have a control of time, because I'm really lost in that VR thing. And at first I was really skeptic of this, because as you pointed out when you were trying to tell me that you should test the system, and I said nothing here cannot be done in like a normal way, like in how we traditionally analyze data, right, in front of that computer. But when you are in this VR world, you get it's not the data itself, it's about you, that you somehow you have a different kind of awareness of your data set, so to speak. So the data are the same, but then the awareness of the data being there, and I like that you can teleport from one space to another, and then have these different tables and these different points around you, and it's a different kind of awareness of data, and it's really nice. You have a different kind of relationship with the data sets that you just see on tables than being with the data, and I can really get loose of track of the time when I'm in that VR system.
So one thing I should point out though is that you can actually do multiplayer mode as well, because we're working in a gaming software, right? So this is actually us in Lund collaborating with some guys in Belgium. So we're both looking at the same data together. So it's been a real boon for collaborative analysis. And we've done this with like 3 or 4 people, and it works flawlessly. So when it comes to true kind of collaborative analysis of data, I think this is really, really cool, and it works fantastically.
The question I have sort of relates to that. It's about workflow. The way you presented that, it reminds me sort of the olden day. You you did some plotting, thought it was funny, you took it along at coffee time and discussed with your colleagues, and then you had a brainstorming session over the plots. So the way you showed that reminded me that you now have in your virtual reality the brainstorming session, and then thereafter, if you got some ideas from your colleague, you would still go back to your desk and try to put flesh on the bone. Is that still sort of the brainstorming session, or is that more or less the final
I mean,
project?
one thing I cannot say that this is the final stop for analysis of single cell data. It can never be that. It will always come down to scripting to produce your figures and to make that script which makes everything reproducible for publication. Okay, so the way I see it is for the initial exploration of the data. Okay, and in our setting it works very well because we don't have someone sitting next to us saying, oh, what does this gene do? What does that gene do? We can throw them into virtual reality for 2 hours and they then come back with more focused questions about their data that we can start working on immediately. So it kind of cuts out that, the 1-2 day period where we don't have to be there necessarily, but they can just do it themselves. And we have bench biologists using this all the time at the moment for that part of the analysis. I can also say that I'm thinking that with Selectfulness and all this other stuff as well, we are starting to build up like quite a decent library of published data as well, and we're now using it as a library for information. So rather than actually going to individual websites of these different producers of data, we're now putting it into one place, so they can just go into the VR room, put the headset on, look at the data, just for 1-2 minutes if they need to, and just leave once they've found out what they need to know. So it has multiple uses, I think.
So essentially what you're doing here is data visualization. How about manipulating a model, like what the example from Watson and Crick? This apparently was the most horrible thing crystallographers ever had to do, was to build a model manually. Right. And when computer graphics came along sometime in the 70s, that was the best thing that ever happened. Right. But it's a different thing manipulating something, an object rotating around, playing with it, than actually building a model to, say, fit a map. So do you actually use things like, maybe that's more a question to you, is the technology to using gloves, manipulating objects and that sort of thing, is it at the same level of maturity as the technology for visualizing things?
So let's get you straight. So when you say manipulating, well, what do you, because what we're focusing purely on is visualization. So I think building models is something which is outside the scope of what we're trying to achieve at the moment. So that's really all I can say. It really is just visualization and trying to break the data down as quickly as possible, not necessarily build things up, I would say.
So you could say that this is essentially the software that you're running, and you could do this with a 1990s SGI with 3D glasses, right? And a controller.
In principle, you could, yeah. It's been a long time since I saw the SGI system, but I guess in principle you could, yeah, maybe.
Maybe I have a comment to this. I think I saw this type of manipulation already a few years ago. In particular in crystallography, where people were shooting ink like atoms in the free places of electron density. Yeah.
Okay, so I think I'd like to thank all of the speakers and participators in the last talk and the whole session again.